Thanks to the organisers for the opportunity to speak. Today I'll be talking about targeted nanopore sequencing and immune escape in malaria parasites. This is a project that was carried out by a talented PhD student in my lab, Mio Now. Human malaria is caused by infection with Plasmodium parasites and transmitted by mosquitoes. Over half the world's population is at risk of contracting malaria, and resistance to current interventions has resulted in stagnating progress against malaria in recent years. So a vaccine is urgently needed, but one's only available, and this is known as RTSS. Based on a single parasite antigen, the circumsporozoite protein or CSP, this vaccine has shown partial and short-lived efficacy against the disease. Malaria vaccine development has been really challenging because parasites have a complex life cycle and hundreds of target antigens. Sequencing the CSP gene in breakthrough infections in a vaccine trial showed lower efficacy against non-vaccine strains, suggesting that diversity within the gene facilitates immune escape. Using publicly available genomes, Mio extracted gene sequences for 23 vaccine candidates and completed population genetic analyses. For CSP, the haplotype network shows that the vaccine strain is rare and a large number of haplotypes are restricted to certain geographic regions. This suggests that RTSS will be even less effective outside Africa. We see this pattern for most antigens. At the population level, malaria antigens are diverse, and this enables immune escape, but exactly which polymorphisms are driving immune escape is unknown. Our solution was to identify immune escape polymorphisms by studying repeated infections in a longitudinal cohort. We combined samples from two cohorts from Papua New Guinea where risk of P. falciparum infection is high. Active sampling was done every 2 weeks, capturing any infections present as well as passively from cases presenting to the clinic with malaria. The data collection included clinical data as well as molecular detection and genotyping. For the sequencing, we used a probe-based capture assay. The probes target 38 genes, and the assay was applied to infections from individuals who had multiple infections in the follow-up period. The Genomics workflow included whole genome amplification followed by enzymatic fragmentation of the DNA. Library prep was done prior to hybridisation with the probes. Samples were then cleaned up using magnetic beads before sequencing on a MinION MK1C device with around 45 samples sequenced per run. Because most variant callers are designed for diploid human genomes, Mio developed a custom pipeline using SnakeMake. This pipeline includes variant calling using both longshot and pepper as well as a machine learning approach, and then merging the results to identify high-quality variants. Once the final call set is identified, it includes phasing of 2 most frequent variants in the sample. Benchmarking was done against 4 other variant callers using reference strain data. The figure in the middle shows the precision of variant calling by Sniper is close to or 100% accuracy above 15x coverage. On the bottom, Mio also analysed artificial mixed infections to confirm the integrity of the haplotype phasing. And on the right, you can see that most of the samples provide average coverage across the targets of at least 50x. Our custom analysis measures variant turnover at individual polymorphic sites. I won't go into too much detail except to say that we identified a small number of immune escape polymorphisms for each antigen. For CSP, we found 8 immune escape polymorphisms, reducing 121 haplotypes down to only 4 haplotypes that potentially represent antigenically distinct serotypes. We can now use this information to measure serotype frequency in the parasite population. Common serotypes can then be further tested in the lab or used to design multivalent vaccines. So in conclusion, we've developed a novel targeted nanopore assay to sequence 38 genes in parallel. Together with our custom pipeline, we generated high-quality sequences for large numbers of samples in a single MinION run. Our pipeline has accurate variant calling above 15x coverage and includes haplotype phasing, enabling analysis of mixed infections which are common in malaria. This approach provides a framework for improving our understanding of infection dynamics, immune escape, and vaccine design. So finally, I'd like to acknowledge the volunteers from PNG who participated in the study, the lab and field workers from the PNG Institute of Medical Research who collected and processed the samples, key people involved in the work, especially Mio, and our collaborators and funders. Thank you.